This is part 2 of the cellular structure lectures, and we kind of got cut off on this last slide. There we go. I was just talking to you about the difference between the cytoplasm and the cytosol. And the cytoplasm is defined as everything between the plasma membrane and the nuclear envelope. Now, in prokaryotes, there's not a nuclear envelope because there's not a nucleus. So the cytoplasm is simply everything inside the plasma membrane. But in eukaryotes, we define the substance that all the organelles kind of are suspended in. It's not a liquidy like water, it's more of a gel. But the gel-like substance is called cytosol. So that cytosol is a mixture, a solution that contains ions, small molecules, and large molecules. So the cytosol is the liquid portion, and the cytoplasm is the liquid portion plus the organelles. All right, and we're going to go through, and even though I already talked to you about every single one of the organelles, we're going to talk about them in more detail. So what you're seeing here is the nucleus. This structure, this whole structure is the nucleus. The nucleus houses the DNA, which directs the synthesis of ribosomes and proteins. The nucleolus produces the proteins. Oh, I'm so sorry. I hope you caught that mistake. The nucleolus produces the ribosomes. The ribosomes then produce the proteins. So that's how that works. Nucleolus synthesizes ribosomes, and ribosomes synthesize proteins. But it's DNA that directs the synthesis of those proteins, that has the code for each protein that determines our traits. All right. We also have endoplasmic reticulum, which is a system of channels, kind of like in a factory, the conveyor belts and different regions of the factory where different parts are made. Well, the endoplasmic reticulum that's shown here is the rough ER, because it has the red ribosomes attached to it. And so the rough ER, since it does have ribosomes attached, its main function is producing proteins. And then it's going to send those to the Golgi complex to be modified even further. Chromatin is all of this purple stringy looking material that you see in here all spread out. That's DNA plus protein. The nucleoplasm is similar to the cytosol of the cell. It's just, a fluid, a gel-like fluid. The nuclear envelope has a double membrane, and there are pores within that membrane that allow some molecules to leave the nucleus. Now, I know you've heard of the term chromosomes before. You may even know that humans have 46 chromosomes, or 23 pairs, one from your mom and one from your dad. Half of your chromosomes coming from each parent. Chromosomes only appear when the cell is in the stages of cell division. So you only see these condensed looking chromosomes when the cell is actually dividing. But other times the DNA is in a form called chromatin, which means that it's really, really spread out. It's not condensed. Chromatin is the thread-like DNA that condenses and forms itself into these chromosomes. This is the chromosome, and this would be the chromatin. Okay. One analogy that I like to give is if you take a piece of yarn, let's just say you're looking at a short piece of yarn, maybe 2 inches long. Okay, so that yarn would be the chromosome. And then if you took all the individual cotton fiber threads or wool threads, whatever the yarn's made of, alpaca hair, whatever it is, you took all those individual threads apart that compose that yarn, that would be the chromatin. So hopefully that helps you understand. Chromatin is uncondensed. Chromosomes are condensed into rod-shaped structures that are held together at their middle. All right, the nucleolus is the structure inside the nucleus that assembles the ribosomal components, the components of the ribosome, called ribosomal RNA. So it basically produces the ribosomes. Ribosomes are composed of a large subunit, the red part, and a small subunit that you can see at the bottom, the blue part. And during protein synthesis, proteins are composed of chains of amino acids. Remember that from chapter 3. So they take the amino acids and assemble them in the proper order to make a protein. And they do this with the help of messenger RNA. It is important that you know that all cells have ribosomes. That means not only eukaryotic cells, which have organelles, but prokaryotic cells have ribosomes as well. What prokaryotic cells don't have are all the other organelles that are enclosed in a membrane. But prokaryotic cells do have ribosomes. The mitochondria, which is found in animal cells and plant cells and the cells of fungi, any eukaryotic cell has mitochondria. And mitochondria is where cellular respiration takes place. And the product of cellular respiration is ATP. ATP is used for energy in the cell. Mitochondria does have its own DNA and its own ribosomes. It is thought that mitochondria actually evolved from bacterial cells, from prokaryotic cells that were engulfed by larger cells. And eventually they lived symbiotically until the prokaryotic cell was just an organelle called mitochondria. Mitochondria have a double membrane. They have an outer membrane, which is just rounded, and then they've got an inner membrane, and that inner membrane is folded and forms these folds called cristae. And the more folds that you have, the more ATP you can make. So the folding increases the surface area in order to give you more ATP. There are some vesicles that do specialized things. Peroxisomes are enclosed by single membranes. They carry out oxidation reactions, which are breakdown reactions, and they specifically break down fatty acids and amino acids and function in detoxifying the body. So you find a lot of peroxisomes in the liver. Glyoxysomes, every time I teach biology, I learn something new because biology is constantly changing and different people write the material. So this is one I've never heard of before until now. Glyoxysomes are specialized peroxisomes that are only found in plants, and they are responsible for converting stored fats into sugars. All right, the endomembrane system is, you may have heard sometime during your life taking biology classes throughout your school that a cell is like a little factory. So the endomembrane system is that part of the cell that's you can compare to a factory. It begins with the nuclear envelope. Here's your nucleus, here's the nuclear envelope. What's immediately attached is the rough ER. And What you cannot see here is the smooth ER, but the rough ER makes proteins and the smooth ER makes lipids. Um, any of those products. So let's say the rough ER has produced a protein, and it's gone through all of these channels here that you see. It's produced this protein, this little green molecule here. That little green molecule is pinched off, in a part of the ER pinches off with it to form a vesicle. And so that's a transport vesicle. Here's one as well. This is a transport vesicle. So that vesicle fuses with the Golgi complex or Golgi apparatus. And the Golgi packages and sorts and modifies that protein. See how it's added something to the end? And a lot of times what it adds is a carbohydrate chain. But anyway, it modifies, sorts, and packages the proteins that are produced by the rough ER. And then that protein pinches off, and it is when it pinches off, it's surrounded by a membrane, so that is a transport vesicle. If the protein has been produced and is designed to be released outside of the cell, then the vesicle will fuse with the plasma membrane and it will release that protein outside the cell. So the endomembrane system modifies, packages, and transports lipids and proteins. The only thing that would be different if we were looking at the smooth endoplasmic reticulum is instead of a protein, we would be talking about lipids produced by the smooth ER. But the endomembrane system includes the nuclear envelope. It includes lysosomes because lysosomes are vesicles that can be produced. So sometimes these transport vesicles that pinch off from the Golgi are lysosomes because the proteins inside are enzymes, and lysosomes contain digestive enzymes. So a lysosome is just a specific type of vesicle. And other vesicles we just call transport vesicles. The endoplasmic reticulum, the Golgi apparatus or Golgi complex, and the plasma membrane. These are the components of the endomembrane system. The endoplasmic reticulum is divided up into the rough ER, which produces and modifies proteins and contains ribosomes attached, and the smooth ER, which produces carbohydrates and lipids and steroid hormones, which are lipids. It also helps in detoxifying medications and poisons, and a big role that it plays is storing calcium ions. Particularly, the storage of calcium ions is extremely important in our muscle contractions and our nerve impulses. So the Golgi apparatus sorts, tags, and when I say tags, it often will add like a chain of carbohydrates or a chain of some chemical to the protein or lipid that was produced in the ER. So sorts, tags, packages, and distributes the lipids and proteins that came from the endoplasmic reticulum. And in plant cells, it can also make polysaccharides, which are complex carbohydrates. You learned that in the last chapter. Vesicles are membrane-bound sacs that function in storage and transport. So we often call them transport vesicles. Vesicles can fuse with the plasma membrane to release their contents outside the cell. We call this exocytosis. We'll talk more about that in chapter 5 or unit 5. And then vesicles can fuse with other organelles such as lysosomes inside the cell. Let's say that a vesicle contains a bacterium or a food particle. If it fuses with a lysosome, the lysosome's digestive enzymes will digest it and break it down. And we'll stop here and begin